Back. So the last thing we're going to talk about, at least in terms of muscle lectures, I will do a little bit of a one with the iPad, looking at some of the actual muscles that you need to know. But the last one we're going to talk about here is really getting into smooth muscle and kind of how it's similar and different from skeletal muscle and how it's going to contract. We will discuss cardiac muscle more in A&P2 when we're actually talking about the cardiovascular system, but this is the other spot where we can talk a bit about smooth muscle. So its shape and how it looks a little bit different. Again, remembering it was kind of wider in the center and tapers out on the ends. Usually has a centralized nucleus, much smaller, much shorter than skeletal muscle fibers. It does have endomysium wrapping around each cell, but it is going to be a single nucleus, not multi-nucleus, and it is also not striated. So you can kind of see an example of it here. You can see, again, centralized nucleus right here, tapering out on each end. And it makes a whole sheet of muscle where they all kind of fit together, something like this. What we're going to see is on the muscle cell membrane on this one, there is going to be calcium channels. There is not transverse tubules. They do have little increases in surface area, these caveolae. And they do not have much of a sarcoplasmic reticulum. What we're going to see on these ones, calcium is going to be important again, but the source of the calcium is not going to be the sarcoplasmic reticulum. It is going to be actually extracellular calcium a little bit more than the sarcoplasmic reticulum. What we're going to see is it is still going to have actin and myosin present, but we are going to see what are called these dense bodies and dense plaques that are connected to the cell in the cytoskeleton, which allows the cell to get both shorter and narrower when it is contracting. And we can see some of the pictures on this stuff here in just a second. These contractile proteins will be arranged in between these dense bodies and dense plaques. We don't have Z-disc, we don't have a normal sarcomere in that sense, and these things are going to be oriented in different angles, and because of that we will not see those striations. Again, still the same contractile proteins with overlapping, but because not all the fibers are lined up in the same direction, we're not going to see those striations at all. And you can kind of see the example right here. You can see these dense bodies and dense plaques. Within the cell, there will be the dense bodies, dense plaques at the surface of the cell. But you can see there's actin and myosin fibers going in between these dense bodies or between a dense body and a dense plaque. When this muscle contracts, you can see that overall the muscle cell is going to get shorter but also narrower, so it's contracting in both of these directions when it's actually contracting. One of the differences on these ones is unlike skeletal muscle, we have myosin heads along the entire length of the thick filament on this one rather than just at the ends. What this can do is this can form more cross bridges. It also means their kind of length power curve is going to be better because we're not limited on those connections between, with it only being on the ends. The other thing we're going to see is these cells are capable of doing what's called a latch bridge mechanism. So what we can do with these cells, and because of how they're activated, which we'll talk about in just a little bit, these cells can contract a little bit, form some tension, and then hold that tension without consuming ATP. And when you think about what smooth muscle is doing some of the times, like in a sphincter, this is a good thing. You don't want lack of ATP to cause the sphincter to open up when you don't want it to. We're going to see these do have actin and tropomyosin, but they don't have any troponin. What they do have inside the cell is something called calmodulin. So when calcium is then going to enter into the cell, you're going to have this calcium bind with this calmodulin and activate it. This then will activate what is called a myosin light chain kinase. When this is activated by that complex, it phosphorylates ahead and it allows activity of the ATPase, which is going to allow those myosin heads to be able to start ratcheting and contracting this muscle. What we're going to see is in order for this to stop, you need an actual something that is going to remove those phosphates. So it's not a kinase now, we're looking at a phosphatase. But to go into relaxation, you're going to have this myosin light chain phosphatase dephosphorylate that myosin head, which will inactivate the ATPase and let the cell go into full relaxation. So again, how are these anchoring filaments put in here? And the contractile protein in this muscle arranged? we don't have sarcomeres. We have these dense bodies and dense plaques, and these filaments are going to be in between those at kind of oblique angles in a number of different directions. The calmodulin and the myosin light chain kinase, you need calcium to bind to the calmodulin to activate that. That will then activate the myosin light chain kinase, which will phosphorylate the myosin heads and allow the muscle to start contracting. So how it really works here, we're going to get stimulation of this muscle. When this happens, we are going to get gates opening up in the sarcoplasm as well as the sarcoplasmic reticulum that are going to let calcium ions flood out into the sarcoplasm. It is going to bind with that calmodulin, forming the calcium calmodulin complex. This can then bind to an inactive form of the myosin light chain kinase and activate it. When it binds to that, it can activate that ATPase, and we can start reducing a power stroke. We're going to get that repetitive power stroke over and over again, these sliding the filaments past one another, and we're going to shorten and narrow that entire smooth muscle cell. In order for relaxation to happen, we need to stop the stimulation. We are going to have to remove that calcium in the sarcoplasm, which is going to allow this to go back into relaxation. The other thing we need for relaxation is you need the myosin light chain phosphatase to phosphorylate these myosin heads and allow these to go back into relaxation. And if we do not activate that phosphatase, what we can do is that latch brake mechanism where it can remain in that contracted state. And I always kind of joke around, I mean, if you think about like your anal sphincter, you want that to stay partially contracted all the time. You don't want it to just go full relaxation on you. And this shows the whole little process right here. So opening of voltage gated channels lets calcium flood in from outside. It binds to the calmodulin, getting that calcium calmodulin complex, which activates the inactive myosin light chain kinase. Once this is activated, it can phosphorylate these heads and lead to contraction. And that whole power stroke reattachment, that whole process. What we're going to see on this one is this is not, if we were looking at a muscle twitch on this one, this is not going to be super quick. There's a much longer latent period between stimulation and the start of contraction. And this is because there's that phosphorylation that needs to take place, and there is some difference in the ATP activity between this, ATPase activity between this and skeletal muscle. The other thing is calcium pumps are not quite as fast, and for everything to kind of go back to normal, the contraction lasts a lot longer because we have to dephosphorylate those myosin heads, and because we can also do this latch bridge mechanism if we do not dephosphorylate these myosin heads. But when you really think about it, you generally don't need a rapid onset of this smooth muscle contraction. It's not something that needs always an immediate response. The other thing is a lot of times we want this to have some type of resting muscle tone, so having that latch bridge mechanism makes sense in the smooth muscle. The other thing is because we can do that latch bridge mechanism, we have muscles that are much more fatigue resistant. Again, you don't want your anal sphincter to get fatigued. So energy requirements are much lower. We can do this almost entirely through aerobic mechanisms. And because we have that latch bridge mechanism, we can maintain a contraction without continuous use of ATP, which means we can keep this muscle contracted for a long time before it will fatigue. The other thing is because we have these myosin heads along the whole length of the thick filament, we don't have a length tension curve as much. We have a much broader length tension curve where this contraction is much powerful throughout the whole length if it's stretched out or if it's really already contracted quite a bit. It is able to contract forcefully in both these ways. So like it says, if there's half the length already contracted halfway or stretched to double resting length, it can still contract forcefully in both those cases because we have these myosin heads all along that filament. And this is important with stuff like the bladder, where if it's getting really full, you still want it to be able to contract and force that urine out. Or if it's not particularly full, you still want it to be able to force that urine out if you're in the bathroom, going to the bathroom. So what is unique about it? Long duration, fatigue resistance, help you maintain that tone, and we have a much better tension length curves to mean that we can get forceful contractions even if this muscle is very stretched out or very contracted already. The other thing we're going to see in this one is this is controlled not via the somatic nervous system, but via the autonomic nervous system. So this is something that we do not have voluntary control over. If you've ever had your stomach growling, especially during a test, you can kind of realize this. These respond to acetylcholine still, but again, this is through the control of the sympathetic and parasympathetic nervous systems. So these ones are generally going to contract in response to acetylcholine, relax in response to norepinephrine. A lot of times these ones will also contract in response to stretching. So a lot of times a smooth muscle, if you stretch it, it will contract immediately and then start relaxing after a period of time if that stretch is maintained. The other thing is that causes us to contract a lot of times is other different hormones. If you have changes in pH or oxygen or carbon dioxide levels, these are all things that can sometimes stimulate these muscles to contract. Oxytocin being an example of a hormone that causes smooth muscle cells in the uterus or in the mammary glands to contract in response to its release. So, and again, autonomic nervous system as well as environmental changes around these cells are really going to be the ways that we stimulate these smooth muscle cells. The other thing we want to talk about is smooth muscle cells sometimes can function in a couple different ways. You can have what is called multi-unit smooth muscle, which you can see is at the top on the right, where each cell kind of acts on its own. So you have these autonomic neurons with these swellings called varicosities that can release these neurotransmitters that will act on each single cell to get it contract. So this is acting more like a motor unit, like we saw with skeletal muscle. So we see this in like the eye muscles, your erector pili muscles, certain walls and arteries, the respiratory system. Again, arranged in motor units where you have these neurons coming in there and these varicosities off that. Greater tension, and again in the smooth muscle as more is stimulated. The other option is this single unit smooth muscle, which is usually digestive tract, reproductive tract, and a lot of other blood vessels. On this one, you have these cells connected by gap junctions, which can once you get a depolarization on these initial cells, it'll transfer through all these, and these actually function as a single unit instead of each cell having to get stimulated separately. If you stimulate one of these cells, it will cause stimulation of all the connected cells. So that's the difference between this multi-unit smooth muscle versus single-unit smooth muscle. Multi-unit smooth muscle to me acts a lot more like skeletal muscle. We'll see single-unit smooth muscle acts a lot more like cardiac muscle actually does. So usually, like I said, that single-unit one, it is going to be stimulated through varicosities. You're going to get synaptic vessels releasing this neurotransmitter, and you're going to get this whole connected set contracting as a unit. Again, we find smooth muscle throughout the body. It's involuntary. It's in blood vessels, in the cardiovascular system. It makes the sphincters and a lot of the tubing of the digestive tract. We find it in the skin for blood vessels as well as the erector pili muscles. The respiratory system, it helps control airflow in and out of the alveoli by causing bronchial constriction or bronchodilation of the bronchioles in the respiratory system. It also is found in the sphincters of the urinary system, within the bladder wall, as well as the ureters to move urine down to the bladder, force urine out of the bladder through the urethra. Also seen in the male and female reproductive tracts in a number of areas. And again, you can look at these ones. We'll talk about this in A&P 2. So really, that's what we need to know is smooth muscle. The last episode I'm going to do on this stuff here is going to be going through the actual skeletal muscles that I need you to know for the body, and what would be generally more of a lab-based thing. I will find an app on my iPad and do, much like I did with the bones, a couple of shows looking at some of these different muscles that I need you to know. So I will see you again next time.